My name is Anusha Kalbasi. I'm a associate professor at Stanford. I'm a radiation oncologist, so I see patients in the clinic, but I also lead a research laboratory and participate in running early phase clinical trials for patients with cancers, solid cancers especially, and I actually focus on sarcomas, which are a rare tumor. So I understand, some of the challenges with finding treatments for rare tumors. But what I wanted to tell you all about today is the trial that we've actually been working on, a clinical trial that we've been developing for about 5 years. And originally, we were developing it for a completely different disease, melanoma. Um, but we have some reasons to believe that patients with neuroendocrine tumors, at least some patients with neuroendocrine tumors, might also be eligible for this treatment. Um, so I have some slides, and I don't want to spend too much on slides because I'd rather have a conversation with you. So just a few minutes, but then we can maybe have some Q&A and hopefully answer some of the questions around this. So this is, first of all, the big group of folks that has helped put this all together. And one of the beautiful things about this project has been its collaborative nature. I started it while I was at UCLA as a faculty member and with colleagues at City of Hope who helped us develop this technology. And then now I'm at Stanford and we've roped the Stanford community into this as well. Um, and the idea is that, your immune system, and especially T cells, these are white blood cells that are circulating throughout your body, actually have the capacity to find and kill cancer cells. And we know this works because there are now drugs out there that can turn on the T cells and allow them to fight the cancer cells in certain cancers like melanoma and some other diseases. But it doesn't work for all patients or all cancer types and hasn't had that much success in neuroendocrine tumors yet. And so what I wanted to talk about was, and I'm going to skip ahead forward, is a new kind of technology that's been around for over a decade now that has cured patients with blood cancers. And this is called CAR T cells, where your T cells would be taken and genetically modified to have this protein on it that allows the T cell now to recognize the cancer cell because of the cancer cell has a special molecule on its surface that allows it to be distinguished from normal cells. And so we can take one patient's T cells and modify them to become a military army against the tumor cells and infuse these T cells into the patients. And the great thing is that they can not only potentially kill the cancer cells, but they can also serve as a living drug within your body and have memory and control any cancer cells that may pop up in the future. And that's the vision for this type of therapy. And I want to say it's not just a vision. This type of therapy has resulted in cures for patients with leukemia and lymphoma where there were no cures before. And so it's been a huge advance, and there's a ton of energy going into advancing these types of treatments for patients with other types of cancers. And so one of the biggest challenges is finding one of these targets on the cancer cells that we can go after. And we found one of these targets for patients with melanoma. And this target also works for patients with certain brain cancers called glioblastoma. And our colleagues at City of Hope had developed this technology for patients with brain cancers and had some promising results. But they allowed us to use that technology to try to treat patients with melanoma. And so we went through a lot of work to get this trial off the ground for patients with melanoma. And we've treated now 5 patients, and we're excited about some of the early results of this trial. But now that we've done this, we started to think, well, are there potentially other patients that might benefit from this technology? And what this graph shows is how high the target is on different cancer types. And you can see there's this one group on the far right over here that's pretty high up there and that I haven't highlighted it. And that's a group of patients with types of neuroendocrine tumors, paraganglioma and pheochromocytoma. And these tumors have very high expression of this target, at least in this data set and some other ones that we've looked at. And so we were intrigued, and we were always wondering about that. But we were focused on developing this for melanoma, so that was kind of um, off to the side. But now that we've built some experience with melanoma, we thought, well, let's explore and see how this might benefit other populations of patients. So at this stage, we want to know, what is the possibility of treating patients with neuroendocrine tumors? How high is this target on the tumors of patients with neuroendocrine tumors? What can we learn about that? And can we potentially enroll patients with neuroendocrine tumors on our study? Our study is open to all cancer types. It's not actually open just to melanoma. We designed it intentionally that way. We've just focused on melanoma so far. But right now, our goal is to expand our reach and to learn about the expression of this target on tumors types other than melanoma. And one of the key targets being patients with neuroendocrine tumors. And so that's why I'm here to talk to you all today and tell you that we have a pre-screening process that's pretty easy to participate in, where if a patient is willing, our team at UCLA will ask the institutions that may have your biopsy material for a few slides of those biopsies to perform some testing to see if this marker, which we call IL-13 receptor alpha 2, if that is expressed on your particular tumor type. And if it is, we gather that information. And you may be eligible if that's something that you and your physicians are looking for additional treatment options. This may be an option for you. However, it also serves as a piece of information for us and for other patients throughout the country, whether or not, what is the chance that patients with, for example, neuroendocrine tumors have this IL-13 receptor alpha 2? Because frankly, the data sets that are out there right now, it makes it really hard for me to give a number. I couldn't tell you if it's 20%, 40%, 90%, or 100%. We were kind of playing a guessing game, but we think we have some good clues that the expression is high, at least in some types of neuroendocrine tumors, but we don't really know. So just gathering this information will be tremendously useful for us. And the cherry on top is it may actually provide a potential treatment option for patients who are looking for that. And so that's where we are, and that's why I'm here to speak with you today to introduce that this trial is not only we're not only doing this testing, and it's free of charge. It's just it's a research test. Really, it just requires a patient's consent by the telephone to allow us to reach out to your, wherever the tissue is stored, whether it's at a hospital or clinic, and ask for that tissue so we can test it, and then we report the results back to you and your physician. So that's really the outreach that we're doing today, and to share about this. But on top of that, tell you that this trial is actually ongoing. So while Lisa said this may help patients down the road for years down the road, we actually have this trial today. And so patients can be screened and treated right now. And so it's kind of an option for both. It could be an option for now. It could be an option for a year from now. But it's important information also for us to learn about the neuroendocrine tumor community. And so that's why I'm here.
Thank you for that. That's really exciting. And if you could also kind of explain a little bit, I know you've shared with me where this is available right now.
It's a great question. So the trial is open at UCLA, and we participate on the trial, when we have to make the cells to treat a patient. The cells have to be made at City of Hope. So patients do have to take one trip over to City of Hope to have their white blood cells collected so that they can be genetically modified. And then they create this product out of your own cells. And then the cells are shipped over to UCLA and the patients are treated. And I'm at Stanford, so my lab runs a lot of the analysis of the biopsies and blood tests that patients undergo during the study. And so the samples are often later on shipped to us so we can do analysis. The goal in about a year is to then open the trial also at City of Hope and Stanford so that patients throughout California and the country have access to all three this trial at all three sites. But at this moment, at least for the next 6 months to a year, it's primarily at UCLA.
That's really helpful. So say someone is, interested in finding out if if their tumors have this receptor, and
Mhm.
if this trial might be an option for them, but they're not ready to commit. What is the commitment level option, and how long would they have to like decide or any of that?
Yeah, there really is no commitment. The only commitment is to allow us to access the samples that you have stored at whichever facility that is. If the test comes back positive, that's your information. You choose to do what you want with that. If you'd like to consider the trial, we are happy to meet with you and discuss that. If you say, not for me right now, we're fine with that too. There's all those options are available. And then if 2 years from now you decide, hey, maybe now is a good time for me because of whatever reason, then we can revisit that, presuming the trial is still ongoing at that point. But we anticipate that we'll be accruing probably for the next couple of years. But likely there will be other iterations of this, presuming that we have the success that we hope we have.
This is really helpful. And you described this to me as pre-screening. So,
Yeah.
many people hear about screening for a trial. So
Yeah.
this is the pre-screening. How's that different?
Yeah, pre-screening means we just want to know if you're eligible or not. We want to know if this protein is even on it because we don't want to have to go through all the testing, the blood tests and the test to make sure your heart is strong enough and the lungs are strong enough and other things, or the scans that you need to do that we would normally do during a true screening. We want to, before we go through all that hassle, we want to know, is this treatment going to be potentially beneficial for you? Is this protein expressed on the surface of your cancer cells? And so it's, yeah, absolutely right. Pre-screening, very different from screening. We're just gathering some information. It's like a survey, but with a small test of your tissue sample.
Yeah, so gathering information. That's part of why in the intro I was kind of saying, well, it can be further down the line. It doesn't have to be
Exactly.
tomorrow at helping
Exactly.
you, but there's a potential.
So a couple things. One is that usually places when you do have a biopsy or a surgery done, facilities keep the tissue for quite a long time, so it's usually there. The And the second point is that the trial's open to people all over the country. So yes, but I understand getting here is a challenge. But for the pre-screening, we actually can do the consent for the pre-screening by telephone. So
Mhm.
there's no need for a visit. Now, if a patient were to be positive and would be interested in doing actual screening, then they would have to come to Southern California to do that. And so that's something you'll have to think about, if the results are positive, what would you do with that information? But that gets back to my other point is that even if we just get this information, that's helpful for the community. And so even if it doesn't necessarily change what you may do in the immediate future, it may help us learn more about the expression of this target on neuroendocrine tumors.
So just to clarify, how fresh does the tumor sample have to be?
It doesn't. These are usually they're essentially always in fixative, so they can last for decades. The more recent tumor biopsy is, I think it's helpful. It gives us a sense of what's going on right now because tumors do change over time. But we don't have a lot of reason to believe that if it's positive a year or two ago, that it would suddenly be gone now. So we would rely on whatever information we could get. The reality is, cancers and tumors can be different. One tumor in one location may be different from another, but we don't have, it's not realistic for us to biopsy every tumor in a patient's body. So we kind of trust the results from one tumor and hope that that's representative of everything at this point. So, but any lesion, whether it's the primary or a secondary lesion, would work. I think right now, what I would want to emphasize is hopefully we can encourage folks to do the pre-screening. And if they're positive, then we can go into the specific details because that also depends very much on each patient's individual situation. But just to give you a big picture, the process is patients come and they have a few days of a chemotherapy regimen to kind of clear some space in the patient's body for the new cells to come into the body. So it's a few days of chemotherapy, and then they receive the engineered, the genetically modified T cells, their own T cells, and that's one infusion, one dose, and then that's it. That's the treatment. And we've had several patients go through with minimal side effects and toxicities, but there are some things that can happen in terms of the immune system getting too turned on or not, or some inflammation that can happen related to the immune system being turned on. And we're happy to go through those details with the patients once they're kind of considering the screening process. And there's actually a lot of literature out there for patients about, if you look up CAR T cell therapy, because it is a standard treatment now. It's an FDA approved treatment for leukemias and lymphomas. So the side effects related to those treatments will be very much similar to the side effects that we would counsel patients on when they're considering the trial.
Will you test only for this specific protein, or can we unlock more potential answers with you looking at the slides, like an actionable mutation?
That's a great question. Our test is very specific for this protein, and so we wouldn't give any other information. We wouldn't be collecting any other information. But there are other tests out there. I'm sure if you speak with your physicians for genetic and molecular testing of tissues that can be done through companies and institutions to kind of unlock as much information as possible. But we're focusing on this because this is the one that we have an actionable kind of next step for. So that's why we're doing this.
Really exciting.
Do any of the standard genomic tests, Strata, there's one from Invitae, Tempus, these groups, would any of those already cover this marker, or are you looking for something new and different?
Yeah, that's a great question. Those tests are typically based on the DNA or RNA within the cells, and that tells you some information, but we're actually looking for the protein itself on the surface of the cells.
Sure.
Yeah, which those tests don't cover. So, your Tempest test may tell you if this gene is on in your cells, which would be actually an interesting clue to indicate. If you don't have the gene, it's unlikely that it's going to be on the surface of the cell. But and not all these tests have the gene levels. So ultimately, we would have to do the protein staining test. But it's a great question.
Thank you. What if someone had a biopsy? Can they use a cytopathology sample instead of I guess, I don't know if you mean the tumor sample. Can you use a smaller tissue sample?
Yeah, we can. And we use biopsy samples a lot. Now, there's some biopsies that are done with a fine needle, which only gets a few cells out of there. And those are not ideal because testing 3 cells, it can be subject to normal error. So the bigger the biopsy, the better. And if it's a surgical specimen, even better. But as long as it's a core biopsy of some type, then it usually will work. Our workflow has typically been having the physicians send a note to our team at UCLA. There's an email with we just need to know the patient's information, contact information, so they can call you and get the consent over the phone. And then the information about where the specimen is and the number of the specimen. And that actually might be easier for the physician to get. And that's why we encourage this to happen through the doctors, because then once the results are back, we can also then contact the doctors to work with the patients. But I think it would be possible for the patient to directly consent. I just think that's been our workflow so far, to have it done through the physicians. The UCLA would be the one figuring that out, and that can be a little bit tricky. It may mean that it will take a little bit longer for us to get the tissue and have it sent to UCLA, and so that's why it's good to do these things earlier, because in best case scenario, it'll take a couple weeks, but worst case, it could take months just to track the tissue down and get it to UCLA and do the staining and get the results back. So yeah. It's certainly doable wherever the tissue is. It just may take a little longer.
Could you describe again what type of nets qualify for this? This is actually an excellent question.
So first of all, any tumor type qualifies for this. We're not really restricting, but what we're trying to focus on is all patients with neuroendocrine tumors. So any neuroendocrine tumors. The reality is we don't know which ones will express it more than others. What we do know, and what I have some experience with, is the paraganglioma and pheochromocytoma patients. We think those patients certainly will have a higher chance of expressing the target. So if, if I would say one particular type, I would say those patients, paras and POs, but I think really we're interested in screening all patients with neuroendocrine tumors.
So if I understand correctly, a lot of this is gathering information, and we can help with information gathering to see what type of neuroendocrine cancers
Yeah.
might have this receptor. And then if you do have the receptor, this might be a potential option for you.
Exactly. And in parallel, we're doing doctors like Dr. Coons and other physicians have compiled, databases of tissue from patients of neuroendocrine tumors that we're testing now so that we can have a little bit more accurate information so I can come tell you, if you have a, pancreatic neuroendocrine tumor, your chance of having this is, 15% or 40% or whatever number it may be. So hopefully in the next even month or two, we'll have that information, and I'm happy to update everyone, because if we find out that, for example, small cell neuroendocrine tumors of the lung don't express this ever, I also don't want to waste folks' time and have you sending your tissue for something that has nearly 0% chance of having it be positive. So as we get that information, I'll be sure to update the community so that folks can prioritize the types that we think are most likely to be positive.
Yes. So that's helpful to, know how this information is being used. Is there a cap on the amount of patients, the number of patients you're going to be enrolling or pre-screening?
Well, the pre-screening, no, there's no cap. Although I will say, if we suddenly get a deluge of thousands of patients sending pre-screening samples, then we may have to narrow the criteria a bit. I don't think that's going to be the issue. But in terms of the actual trial, this trial is a phase 1 trial, so it's not a huge trial. But we expect to enroll about a total of 18 patients, so about 10 or 15 more than what we have now. But as I said, this would be the first iteration, and if we see signs of success, we'll likely do a follow-up trial. So that I wouldn't say that should be a major concern at this point.
And is it any help for someone who has no evidence of disease right now that doesn't need to participate in a trial to also submit these tumor samples?
I mean, I think we would prioritize patients who think that they may need a treatment in the relatively near future. So I think that would be our emphasis. But if there are patients who want to contribute to this, we're always happy to test, and it does provide us some information. So certainly happy to accept that.
So I'm wondering, this is really interesting to me. This is really exciting because, a lot of people have asked about immunotherapy for neuroendocrine cancers, and there's a lot of work to be done. We know that Penn has a trial that they're going to be opening in CAR T, and there has been work done at Moffitt. How does this compare to other CAR T trials that are out there?
Yeah, there are lots of different flavors of CAR T cells, and the differences are what are they targeting? What is the molecule on the surface of the cell that's being targeted? So the different CAR T cells will target different types of proteins on the surface of cancer cells. So that's one major difference between different CAR T cell types that are developed at different places. And then each CAR actually, there's lots of it's kind of like a boutique therapy, because each place will make their cars in their own way. They have special designs for the editing of the DNA. So there are specific characteristics of every CAR T cell product, but the principle behind all of them is the same. And so I think I can't answer that in any more detail unless I know exactly which CAR T cell we're comparing against. But suffice it to say that the principles of how CAR T cells work is the same across different centers. It's just what are they targeting, and what are the kind of boutique considerations of each center. So that's something that we can help you navigate once we know if you express this target or not, and we know what other options are available to you.
Okay, so this is an opportunity to find out what type of tumors have this target, find out if our particular tumors have this target. So this trial might be open to us.
So we
Yeah.
did
I would approach that. I think that if you have a good relationship or active relationship with your oncologist, I would let them know about this and let them know that it's pre-screening, that it's not actual screening for the trial. Because I think, um, again, we would, whether depending on if you're positive, we would then obviously have a discussion with your oncologist on whether this would be something to consider. And we don't want your oncologist to think that, you're going to enroll on another trial. That might raise some alarms. So I think the important thing is to say this is just a pre-screening, just to know if your tumor expresses this target and you like to participate, and ask them to refer you to the trial. Now, if you have any pushback, we're happy to take your direct information, and this information is here. But I think in general, our workflow has been to go through the physician, so we'd like to stick with that as much as possible.
This is all really helpful. So have our physician contact the UCLA team, talk to our physician if we're interested about pre-screening, now that we're going to change treatments in the middle of the course.
Yeah. As an oncologist, I know that we all feel ownership of our patients and we want the best for them. And there are also a lot of trials, and not everything is the right fit for a particular patient. And your oncologist knows your situation the best. So if one of my patients came to me and they were saying they were going to go on another trial, I think initially I would say, well, let's see what trial this is and see if it makes sense for you before you go, or you may not even need this right now. So I just don't want to raise alarms with the oncologists. And it may just, they generally want to know because they want the best for their patients. So I don't think it'll be pushback. It's more just making sure everybody understands, the situation.
Got it. Thank you. I feel like this opportunity is really exciting. I think our group is so pleased that you came to talk to us today to give us a chance to do some research and open the doors to a possible treatment for us. So thank you very much.
My pleasure. And I know there are actually a few other efforts in this area with neuroendocrine tumors. So I hope this is the first of others. Yeah, I'm really excited to learn more about how this target might be used for patients with neuroendocrine tumors, and we hope that we can help some patients soon.
I found that the key was, as you have highlighted, using this pre-screening language and talking to our oncologist. And then when we brought it up, as a researcher himself, he was excited. Of course, he wanted to know what kind of trial this was, but understanding that, as he explained to us, this contributes to the future of what might be an open door for you, tomorrow, next month, in the future. But, things take a while too, and there's not a commitment to it. And he was really excited about this overall landscape. But yeah, not necessarily like taking a detour from what we're doing today, but opening doors for possibilities.
Exactly.
This has really been super helpful, really interesting. You can see this group really likes to learn. Everyone likes to know what's out there, and to know what's on the horizon too, for the field of NETs. What people like you are working so hard to do for us. So we're really grateful for it. Of course, we want to know how it applies to us today, but also seeing the big picture helps, because what helps other people helps us as well. So thank you for all that you do on behalf of the NET community. I mean, really dedicating so much time, energy, research to this really means a lot to us personally, and it gives us a lot of hope. Thank you.
Thank you for what you guys do, and this group. I think these kind of thought centers and patient support groups are so important, and they help us figure out, how to accomplish these goals that we have to help patients, because we can't do it in isolation. So, if this group didn't exist, it would be hard for me to figure out where to go to advertise to patients with neuroendocrine tumors. So thank you for all you do.